Our next talk is from Dr. Koji Kanayama, and he's going to be speaking on the influences of oxygen tension on human dermal papilla sheath cells in hair regeneration.
Good afternoon, everyone. My name is Koji Kanayama. It's a great pleasure to be able to speak to you. My presentation today is about influences of oxygen tension in expansion culture of human dermal papilla and dermal sheath cells on hair regeneration. There is no conflict of interest. Introduction. It is well known that hair-inducing capacity of dermal papilla cells called DPCs and dermal sheath cells called DSCs is easily lost once cultured. Optimized method to cultivate DPCs and DSCs is needed for establishing cell-based human hair regeneration therapy. In this study, we focused on oxygen tension. Conventionally used 20% of oxygen concentration provides very high oxygen tension to cultured medium compared to in vivo physiological environment. We hypothesize oxidative stress by hypoxia or anoxic stress by hypoxia are harmful to DPC and DSC culture. The purpose of this study is to investigate whether normoxia helps to maintain their original functions compared to hyperoxia or hypoxia. Primary culture of human DPCs and DSCs were performed. For proliferation assay and PCR, cultured cells were put under hypoxia, normoxia, or hypoxia. For microarray analysis, we compared cultured cells under hypoxia with normoxia. In vitro, gene expression analysis was performed. In PCR, we analyzed messenger RNA expression of hair-inducing capacity-related genes. In the microarray, we compared gene expression profiles of cultured DPCs and DSCs under normoxia and hyperoxia. In vivo, hair follicle reconstitution assay was performed. We transplanted cultured human DPCs and DSCs mixed with newborn mouse epidermal keratinocytes into the back of the nude mice. 8 weeks later, reconstituted skin samples were collected and histologically evaluated. This is the result of proliferation assay of DPCs. On day 5 in logarithmic growth phase, cell number is highest in normoxic group. Hypoxic group was inhibited for cell proliferation. BRD assay showed ratio of BRD positive proliferating cells was highest in normoxic group. This is the result of proliferation assay of DSCs. On day 5, cell number is highest in normoxic group. Hypoxic group was promoted for cell proliferation compared to hypoxic group. BRD assay showed ratio of BRD positive cells was highest in normoxic group. This is messenger RNA expression level of hair-inducing capacity-related genes. We found there are different patterns of expression between DPCs and DSCs. In DPCs, alkaline phosphatase, TGF-beta2, and Wnt10B were highly expressed compared to DSCs. On the other hand, in DSCs, BMP4 and SOX2 were highly expressed compared to DPCs. In DPCs, several kinds of gene expression dropped according to oxygen tension. In DSCs, expression level of BMP4 and SOX2 were highest in normoxic group. We checked the relation between culture time and gene expression level of DPCs. Many kinds of gene expression were dropped according to culture time, especially expression level of ALPL, BMP4, SOX2, and Bashkiran easily dropped in hypoxic group. This is the result of gene expression profile of DPCs and DSCs. We compare normoxic group with hypoxic group and listed the ratios of gene expression level. Upregulation more than 2 times was shown in red characters, and downregulation less than half times was shown in blue characters. As for hair-inducing related genes, there were no special difference in DPCs, but in DSCs, BMP4 expression was upregulated, and alkaline phosphatase and TGF-beta2 expression was downregulated. As for pluripotent marker genes, in DSCs, a lot of gene expression were upregulated. We performed hair follicle reconstitution assay. In positive control, dermal cells of newborn mice were transplanted. 8 weeks later, visible hairs were observed in the transplanted site. Physiologically mature regenerated hair follicles were also observed. In negative control, dermal cells of adult rat soul skin or newborn human horse skin were transplanted. Hair induction were rarely observed. We transplanted cultured human DPCs. Visible hairs was not observed. Physiologically, the number of regenerated hair follicles per sample was slightly highest in normoxic group, though most of hair follicles were immature. We transplanted cultured human DSCs with DPCs. Visible hairs was not observed. Physiologically, the number of regenerated hair follicles per sample was slightly highest in hypoxic group, though most of hair follicles were immature. The maturity of hair follicles tends to be high in hypoxic group. Normoxia enhanced proliferation of DPCs and DSCs. Hypoxia inhibited proliferation of DPCs, but enhanced that of DSCs. Hyperoxia inhibited the proliferation of DSCs. In DPCs, alkaline phosphatase, TGF-beta2, and Wnt10B were highly expressed compared to DSCs. In DSCs, BMP4, SOX2, and some pluripotent marker genes were highly expressed compared to DPCs. Hair follicle reconstitution assay showed that normoxia promoted hair induction in DPCs and DSCs. Hypoxia promoted hair induction in DSCs. Optimized oxygen tension for expansion culture is different between DPCs and DSCs. Although normoxia is better than hypoxia for culturing DPCs and DSCs, DSCs seem to prefer lower oxygen tension compared to DPCs. Application of physiological oxygen tension is appropriate for expanding DPCs and DSCs with preserving their original characteristics compared to conventional high oxygen tension. Thank you.
Thanks, Dr. K. Any questions for Dr. K? So how did you modulate the, I can ask you a quick question. Did you modulate the oxygen tension just through different chambers? One comment is that I think the dynamic range for your in vivo hair reconstitution assay is not very big. It's really semi-quantitative, so it's worth being careful about having conclusions. But great talk. Okay, well, I think this concludes the session. Thank you so much for everybody's attendance.